start now. It's my pleasure to welcome all of you, friends, alumni, colleagues. A very special welcome to this special Mason Science Series event. My name is Fernando Morales Wilhelm, and it's my honor to serve as Dean of the Mason College of Science for all of 15 weeks now, almost. There's been tremendous and appropriate focus on the trials and challenges we've all been facing during this COVID pandemic. The challenges we're facing personally and as well as being the largest public university in Virginia are very real. But I would like to say just a few words about some of the more inspiring experiences of the past few months. Our faculty and staff in the College of Science and across Mason have stepped up. They have worked tirelessly to revamp courses, learn new teaching technologies, support our students, not only in the classroom, but also in our research environments. Many of our researchers, including the ones you'll hear from this afternoon, quickly pivoted their labs and their technologies and their experiments to develop diagnostics and treatments for coronavirus. Our scientists are collaborating with scientists around the world to meet this pandemic with tested, effective solutions head on. At Mason, we've applied a scientific approach to what we call our safe return to campus. So far, with nationally recognized success, our COVID infection rates as a university are among the lowest in the country. Many of our College of Science faculty served and led the task forces that developed the university's protocols, which, by the way, are assessed and challenged daily in ongoing efforts to produce even better results. And you know, that's what science does. Science solves the world's biggest problems. As we get to know one another, you will hear me say this repeatedly. Never before has impactful science been so important. And truly impactful science is what we're going to learn more in this session. Before I introduce our scientists, I wanted just to go over some Zoom housekeeping items. I think first, this event is being recorded and a link will be shared and posted on our website after the event. Second, questions can be submitted. Your name will be anonymous at any time through using the Q&A box for the discussion session following the presentations. And we're going to keep attendee microphones, cameras, chat, and raise hand features disabled for the event just so we can keep things going. October is Breast Cancer Awareness Month, and our scientists in the Center for Applied Proteomics and Molecular Medicine, or like we call it, CAPMM, are leading the world in groundbreaking research and treatments for this tragic disease. Dr. Lance Leota and Dr. Emmanuel Petricoin, or as we refer to him dearly as Chip, are co-directors of CAPMM, which was established at Mason in 2005. Lance holds an MD and a PhD in bioengineering from Case Western Reserve University. He previously served as chief of the Laboratory of Pathology at the National Cancer Institute, NCI. He was deputy director of the NIH, director of the FDA clinical proteomics programs. He holds more than 100 patents and is co-author on over 700 publications. Chip earned a PhD in microbiology from the University of Maryland and previously served as co-director of the FDA NCI clinical proteomics program and as a senior investigator within the Center for Biologics Evaluation and Research at the FDA. Chip is co-founder of 4 life science companies, holds over 40 filed and published patents, and is co-author on more than 400 publications. They are globally respected scientists and successful entrepreneurs who have translated their science into real-world problem-solving and life-saving applications. I would add to these impressive accolades their passion for their work and their commitment to nurturing young scientists. It is really a pleasure to walk, as I did recently, into Chip and Lance's lab and find such a vibrant, diligent working atmosphere these days. It's really uplifting. I had the experience of doing this myself. So I'll ask Chip and Lance to join me now and provide an overview of their work and introduce their colleagues who will discuss the research and clinical trials in more detail. And following the presentations, we'll discuss questions submitted by our audience. So feel free to submit those questions by the Q&A as the presentations are underway. Chip and Lance, welcome. Please give us an overview of the research expertise and technologies developed by CAPMM and introduce your colleagues.
Thank you, Fernando. Really a pleasure to be able to be able to speak today. And Lance and I will be giving a general overview first of our center, but we're not going to take too much time up for that because of all the exciting research that our three senior investigators in our center will be telling you about. So we want them to actually share the spotlight. So our Center for Applied Proteomics and Molecular Medicine is really focused on studying diseases and translating discoveries for patient benefit. We have one of the few laboratories in the United States that has a CLIA CAP accredited laboratory component to our laboratory where we can translate important findings to the bedside. We've already heard a little bit about Lance and I. We don't need to take up any more time going through that, but we have a great collaboration because of Lance's background as both an MD and a PhD in bioengineering, and my background at the FDA and as a cell signaling biologist. We've been able to work in a great partnership for many decades now, and the senior scientists you'll hear about today have been with us very long time, an essential gang of great minds in our center. Next slide, please. So our expertise really is one that traverses the bench to bedside ecosystem, addressing cancer and other debilitating diseases with expertise in proteomics, which is beyond genomics. Genomics in some ways is not old-fashioned, but it's a view that we're actually rapidly accelerating from, because almost all of the FDA-approved drugs that you take, their mechanism of action is actually against proteins, not genes. Proteins, as we say, the DNA is the building block, but it's the proteins that do the work, and they're the actual drug targets of the cell. Almost all the biomarkers that you get measured when you go to get your yearly physicals, many of those blood markers are proteins, not genes. So proteomics is actually part of our lives. We don't even recognize it. We have experts in proteomics in our laboratory, experts that are medical technologists. We have expertise in pathology, cancer research, and molecular diagnostics. And as I said, we are CAP accredited. Next slide, please. We have a research portfolio that really is, again, focused on proteins. You're going to hear in these three separate talks the importance of measuring not just the proteins, but how the proteins work and communicate in the cellular circuitry as we see. These form pathways inside the cell, and just like your GPS map on your car telling you the direction on which way to go, the way that the cell communicates internally and to its external environment are by and through proteins. These are the drug targets. They are very often the biomarkers for early detection. And of course, their identification of these contact points between proteins are novel therapeutic opportunities as well. Next slide. We have a large number of staff-invented technologies that we've been proudly able to develop and wholly invent in our laboratory. New types of tissue fixatives to new ways of capturing cells to help the pathologist in molecular analysis, new types of technologies to be able to look at the cellular circuitry and be able to tell which patient should get which drug, new types of nanotechnology that act like biomarker vacuums and allow us to detect disease even earlier, and brand new technology of protein painting as an example, which allow us to identify new ways and we can identify drug targets and new ways to interact or disrupt important interactions. Say with the COVID virus with its receptor, we're working on these types of problems in our lab every day. Next slide, please.
And I'll step in now. I'm really honored to be part of this wonderful symposium. And our CAPMM group, not only do we want to discover basic mechanisms of disease in the laboratory and then take those discoveries all the way to patient benefit in clinical trials, as you'll hear about, we also want to give back to the community. And so we have two programs I want to briefly tell you about that are ways for us to have outreach of science from the College of Science to the community around us. Our Aspiring Scientist Summer Internship Program is free for students applying, and they get to work in the laboratories of top scientists doing hands-on science, and then they present their scientific discoveries at the end of the summer. We've been doing this for 14 years. We had 1,000 applicants in 2020. This is directed by Dr. Andrea Cobb. And next slide, please. We have 439 alumni that have obtained all sorts of patents and awards and have gone on to great careers. We've been following them for 14 years, as we said, and we're very proud of the graduates of this program. It's our way of inspiring the scientific talent of the future. Next slide. We also host the Galileo Science Cafe, which is Science Night at Mason, by the community, for the community. And so the community around us brings up topics that they want to hear about, and we get the top scientists to talk about them, but we also maybe have celebrities like a football player or a cancer patient, and we have exciting role models as well as the latest truth about science. The goal is truth and hope about any scientific topic that the community wants us to talk about. Next. And we are very thankful for all the sponsors and granting agencies that have supported us over the years. We've been continuously funded since we came to Mason by NIH and other government granting agencies, but I want to make a special thanks for the Beck Foundation, which has supported our ACIP program and the cancer work that you'll hear about, and the Side Out Foundation, which you'll hear about, which is a unique charitable organization dedicated to breast cancer. We're very proud to have funding by them. And the I Spy program that Dr. Wolfkuhl will tell you about is another groundbreaking, innovative program that support our clinical trial work. Next. So just to review the seriousness of breast cancer, 1 in 8 women in her lifetime will develop potentially lethal breast cancer. And in this year, 2020, 276,000 women will be diagnosed with breast cancer, all potentially lethal breast cancer, with 50,000 new cases of premalignant breast cancer this year. Also in 2020, it's predicted 42,000 women will die of breast cancer, and there's 3.5 million women in the USA who have breast cancer today. This is a really urgent problem, and so we need science to guide us to improved ways for preventing breast cancer so that it never happens in the first place, for treating it once it's diagnosed and detecting it early, but also for personalizing the medicine because each patient may need their own special therapy tailored to their tumor. Next. Breast cancer begins by an overgrowth of cells in the milk duct. And then next, the cells invade out of the duct and infest the entire body by entering the bloodstream, traveling to different parts of the body, and setting up secondary colonies called metastases. Once those metastases start growing, the prognosis is very, very poor. So I'm going to tell you about what the topics are covered by our three esteemed scientific colleagues who are really soldiers in the war against breast cancer. And they'll tell you about their promising clinical trials. Next, please. Dr. Espina will describe an innovative prevention treatment for stopping breast cancer before it starts by killing the early stage buildup of cells inside the milk duct that are destined to become the lethal metastasis. Next slide. Then Dr. Wolfkuhl will describe how she can map the circuitry of a patient's stage 2 or stage 3 breast cancer and use this to determine new treatments, new drugs for breast cancer, but also to individualize which drugs are going to work the best for that patient. Next. And thirdly, Dr. Marilena Parabon will describe the world's first personalized therapy trial for metastatic breast cancer supported by the Side Out Foundation. And this is a trial that personalizes which are the best drugs to give a patient who has advanced stage 4 metastatic breast cancer for which there's no real hope for how to treat them. All three are very exciting, innovative new approaches. Let me introduce our first speaker now, who's Dr. Ginny Espina, and she's an associate professor and she's director of the CAPMM certified lab that you heard about, which she built from scratch when we came here to set up our center at Mason. She has a master's degree from Hopkins and she has her PhD from Mason. Take it away, Ginny.
So my topic is preventing breast cancer before it starts. So the first question I'd like to ask everybody to think about is, why are breasts important? Next. I'm sure everybody has their own opinion, but from a biological standpoint, breasts provide nutritive and immune functions. Breast cells secrete proteins, water, minerals, and antibacterial enzymes. One theory postulates that breasts evolved from sweat glands into these complex tissues composed of a convoluted system of milk ducts with fat, lymph vessels, immune cells, and connective tissue. And at puberty, as shown in this mouse model, mammary gland invasion commences in preparation for pregnancy and lactation, followed by involution after weaning. So invasion is a perfectly normal process in breast physiology. Next. However, when this normal process of invasion and involution goes awry, breast epithelial cells begin to accumulate within the milk duct at pathologically defined stages, which are ductal hyperplasia, which is just more cells than normal, atypical ductal hyperplasia, in which there are more cells, but the cells also start to look differently. They take on abnormal characteristics. The third stage is ductal carcinoma in situ, in which the cells have hallmarks of cancer. The fourth stage is DCIS with microinvasion, in which a few cells are starting to break out of the milk duct. And the final stage is an invasive ductal cancer. A breast duct that has, at the DCIS stage, can be 4 times the diameter of a normal breast duct. And this brings up 2 questions. The first one of which is, at what stage of this accumulation of cells within the milk duct do the cells possess the ability to invade? And if we figure that out, can we prevent breast cancer from becoming invasive? Next. To answer this question, we had to start with the simple process of, can we even grow human DCIS cells in the lab? This had never been done before. Next. We established a workflow between the patient, surgery, pathology, and the research lab. The breast lumpectomy specimen shown here, it has red and yellow and blue coloration to it. That's just ink that was applied by the pathologist to provide an anatomical orientation to the specimen related back to the orientation in the patient. And within this workflow, we were able to confirm the diagnosis of DCIS to assure a sense of quality control, but we were also able to cut the breast tissue into tiny pieces and actually grow them as organoids in the lab. Next. Next. So these organoids spontaneously formed spheroids and duct-like structures shown by the blue arrows from many different patients. And because of my background in the clinical lab, I knew we had to do this over and over again to show reproducibility. And this was a reproducible finding in many different patients. Next. To definitively demonstrate that these spheroids were tumor cells, we used a NOD SCID, a non-obese diabetic severe combined immunodeficiency mouse model. To confirm that the mouse lesion that we saw growing from the spheroids was actually a tumor and not just inflammation, we stained a tissue section with an antibody to human-specific cells called EpCAM, which is epithelial cell adhesion and activating molecule. EpCAM is shown here in green, proving to us that this was an actual tumor and not just an inflammatory reaction in the mouse. Next. The next question is, after we grew these cells, what molecular defects allow DCIS cells to thrive in the milk duct? Next. We know that genetic alterations are a hallmark of cancer cells. So using the Illumina 300,000 cytosnip bead chip, we were able to do molecular karyotyping or genotyping on the breast cancer tissue, the spheroids, the 3D structures that grew out. And what this chart shows you is each line represents a chromosome, and the color of the line represents the genetic alterations that we found. Red is a homozygous deletion, meaning you're missing 2 copies of that gene. Orange is a hemizygous deletion, you're missing 1 copy. Blue is a amplification or duplication of a gene. And green is a copy neutral loss of heterozygosity. Green is considered a neutral alteration and doesn't affect the carcinoma or the genetic status of the cells. Next. And as Dr. Leota and Dr. Petricoin emphasized, proteins are important. Why are they important? Well, as my daughter told me when she was 8 years old and she saw me working at home and asked what I was doing, she said, I get it. Proteins are the verbs of the cell. They do all the work. And so studying the proteins by reverse phase protein array technology allows us to quantify the amount of a protein and its post-translational modifications in a given tissue, a given sample for a given patient. Using this technology, our breakthrough discovery was that the DCIS spheroids are addicted to autophagy pathway for cell survival. Autophagy was not active in the epithelial cells or the cuboidal cells that also grew in our organoids. Next. So what is autophagy? Autophagy means self-eating. It's a perfectly normal digestive process in the cell. And autophagy occurs when a cell is under stress. It occurs when there's a lack of nutrients. Many of you may be upregulating autophagy right now if you're hungry. Autophagy allows cells to essentially eat snacks to make energy in the crowded milk duct. Next. Can we use a drug to prevent invasion or decrease the growth of the cells? All of our findings in the lab are only useful if we can actually translate this information to a patient. And is there a drug, more importantly, that's safe and effective for patients with DCIS? Next. So during autophagy, this cartoon shows the milieu of what is happening in the cell. A double membrane vesicle called an autophagosome forms, and it gobbles up proteins and extra organelles, and it shuttles them to the lysosome where they're digested, and the digestion then creates building blocks, amino acids, proteins that can be used further for cell survival. What we found, though, was that the cells, the DCIS cells, are addicted to this pathway. It's not cyclical in DCIS cells. It's always turned on, so these cells are always finding a way to survive in the hypoxic, nutrient-deprived milk duct. Next. So what can we use as a treatment or as a way to inhibit autophagy? Well, chloroquine, a drug that's been in the news because of COVID, is actually very effective in inhibiting autophagy. Chloroquine has been used in millions of people worldwide to treat malaria, lupus, and rheumatoid arthritis. Chloroquine has a very well-known toxicity and safety profile. And importantly, it's absorbed by the cell and transported into the lysosome, where it increases the pH, interfering with digestive enzymes, very similar to taking an antacid if you have an upset stomach. Next. And to ensure that we weren't inducing any further genetic abnormalities with the treatment, we treated the DCIS organoids growing in the lab with chloroquine and repeated our molecular karyotyping. The surviving cells post-chloroquine treatment had fewer deleterious genetic mutations compared to the untreated spheroids. Next. To translate these findings to the clinic, we established a phase 1/2 clinical trial with Dr. Kirsten Edmiston at Inova Fairfax Hospital. The participants received either the standard malarial prophylaxis dose of chloroquine, which is 500 milligrams, or half that dose once a week for 4 weeks prior to their standard of care surgery. We analyzed by immunohistochemistry, tissue organoids, cytogenetics, and reverse phase protein arrays, the tissue before and after treatment. And this trial design allows us to assess the efficacy of treatment in a very short time period, only 4 weeks, rather than waiting for years for a typical preventive or treatment trial. And the unique aspect is each patient serves as their own control because we measure the tissue before and after treatment. The purposes of this trial were threefold. One was to test the hypothesis that by inhibiting autophagy in DCIS, we will reduce the capacity for DCIS to invade. The second was to test the safety and efficacy of chloroquine in DCIS patients because this hadn't been done before. And third was to evaluate whether chloroquine would reduce the DCIS lesion size as judged by MRI and histology, and if it would suppress the proliferation of the cells. Next. We only had 12 participants enrolled in the PINC trial due to a series of difficulties in recruiting patients. However, even though this is a small and underpowered study, 75% of the patients showed a reduction in the size of their DCIS lesion and a reduction in the proliferation index. The remaining 25% had a stable lesion size, but also had a reduction in the proliferation index. And so this PINC trial has provided data and methodology for additional clinical research with other compounds, either alone or in combination with chloroquine. Next. And I'd like to leave you with a thought. Imagine a future in which we could prevent the emergence of ductal carcinoma in situ by administering chloroquine or maybe another compound periodically to inhibit emerging cancer cells. Thank you. Our next presentation is by Dr. Julie Wolfkuhl, my esteemed colleague of the past 17 years. Julie has a PhD in biological sciences from Purdue University, and she's been doing breast cancer biomarker discovery for 20 years. The past 15 years, she has been doing this work at George Mason University.
Okay. Next slide, please. So today I'd like to start off by sharing with you some background. The work that we do in our lab is sort of based in the knowledge that DNA serves, or your genes serve as the blueprint or a guide for the cell. And then they provide a template for which leads to the generation of proteins. And as Chip stated in his introduction, the proteins are the real working machinery of the cell. And he also commented that many of these proteins, in fact, serve as drug targets, and that many drugs approved by the FDA are, in fact, targeting proteins in your cells. In fact, proteins can even serve as the drugs. And you've all seen commercials for drugs such as Humira or Opdivo on television. Those, in fact, are protein drugs that target defective proteins in our cells. Next slide. So we know through decades of research by many, many labs that cancer is in fact driven by hyperactive or defective protein signaling pathways in cells. There are mutations that occur in genes that encode proteins that are involved in cellular signaling pathways, which are very critical in the cellular communication and signaling networks that allow the cells to interact with their environment and with one another. And when these genes become mutated, they produce proteins that are either non-functional or have dysregulated function. And over the years, milestones such as the sequencing of the human genome, as well as the development of technologies that allow us to broadly profile both the gene expression as well as the protein expression, like the work we do in our lab, has really broadened our understanding of tumor biology at a molecular level. And this has led to the development of many new targeted therapeutic agents that has really revolutionized cancer therapy in the past 20 years. And a perfect example of that would be Herceptin, which I'm sure many of you have all heard of. Now, all of this understanding and the development of these targeted therapeutics that are highly specific begs the question of how do we know which patients will respond to which drug? We know that each patient's tumor at a molecular level is unique. And so this brings us to the concept of molecular medicine, which is really based on the analysis of individual tumors at a genetic and a genomic and as well as a proteomic level to generate information that can be used to select the most appropriate therapies for tumor treatment. Next slide, please. Now, our lab has pioneered the development of broad-scale proteomic profiling of tumors. And we use a special technology that's called LCM-assisted reverse phase protein microarrays. This allows us to identify and measure the activity of drug targets and their associated signaling pathways in individual patient tumors. We isolate tumor cells and enrich for them using laser capture microdissection technology. And then we are able to lyse those cells into detergent-based solutions and print them onto a solid substrate that then allow us to assess the activity of hundreds of different proteins that are known to be involved in cancer tumor growth and development within the cells. And we can do this also across many hundreds of patients simultaneously. Now, in the context of clinical trials, how can we use this technology? This type of information can be used to assign patients to therapies that might be beneficial. And my colleague, Mary Elena, will speak more to that point in the next talk. This information can also be used in a more research element of a clinical trial to correlate with both response and outcome data that allow us to identify potential biomarkers that could be used in the future for selecting treatment for patients. Next slide. So I want to share with you the work today that I've been doing with the I-SPY 2 trial over the last decade. As I said, this trial has been open since 2010, and it is a phase 2 neoadjuvant trial for locally advanced stage 2 and 3 breast cancer. Neoadjuvant refers to the fact that patients receive chemotherapy prior to having their tumor, any residual tumor, removed at surgery. Tumors are locally advanced, meaning that they must be greater than 2.5 centimeters to be included in this trial. And all patients are also deemed to be high risk for recurrence. That's based on a genomic test called MammaPrint. And all standard analyses for both hormone receptor status, such as estrogen receptor and progesterone receptor, as well as HER2 protein status, are performed. And these are standard assays that are usually done when women are diagnosed with breast cancer. Based on those biomarkers of hormone receptor status, HER2 status, and MammaPrint, these patients are assigned to one of 8 subtype categories prior to being randomized to targeted therapies. There are up to 5 experimental targeted agents available for randomization at any time. And the primary endpoint of this trial is pathologic complete response, which means no evidence of tumor at the time of surgery. And that's determined roughly 5 to 6 months after chemotherapy has commenced. Next slide. So the clinical goals of this trial are to identify which of those 8 biomarker subtypes that patients get divided into will respond well to various experimental agents. And one of the aspects that makes I-SPY 2 very unique is that it uses special statistical modeling to randomize their patients. And what happens is that as patients come into the trial and they have treatment, they go to surgery, their outcomes and their subtypes are then immediately incorporated back into these statistical model that then assists in randomizing future patients to treatment arms where those patients that have the same subtype as they do are performing very well. And so the ultimate outcome of all of this is that the better therapies, the better targeted agents that we are studying in this trial will move through the process more quickly and will have greater exposure to patients that have responding subtypes. Next slide. So in the past 10 years that this trial has been opened, 19 different agents have been, and combinations have been evaluated. And I'm going to share with you some data from one of the earlier experimental arms of the trial. Next slide. So in addition to the clinical goals of this trial, there is also a very large research and biomarker discovery component that goes on behind the scenes in the I-SPY 2 trial. And that's where our role comes into play. All patients that enter the trial have both genomic and proteomic profiling of their tumor tissue samples done and completed to support our research efforts. And most of our work to date has focused on the biopsy that is collected prior to the beginning of therapy. And to date, we have a data set that includes more than 750 patients across 7 treatment arms of this trial. We have the signaling information that has allowed us to do a variety of different analyses to identify potential biomarkers. Next slide. So what I want to do is just show you an example of the type of results that we can obtain from this. And we're going to focus on one specific subtype of breast cancer that is included in this trial, as well as one agent that was used to treat these patients. So triple negative breast cancer is a subtype of breast cancer that does not express estrogen, progesterone, or HER2 receptors by standard assay methods. This subtype represents roughly 15% of all breast cancers that are diagnosed. While they are generally sensitive to standard chemotherapy regimens, they have a very poor survival prognosis, and they often relapse within 2 or 3 years of starting treatment. This subtype of disease is very heterogeneous from patient to patient at the molecular level. And unfortunately, there are really no effective targeted therapies available today for these patients. Now, one of the drugs that was included in the I-SPY2 trial that we analyzed was neratinib. And neratinib is a drug that targets the HER family signaling pathway in breast cancers, which is a very well-studied and well-known pathway in breast cancer. And it targets the HER2 receptor as well as the EGF receptor. And we hypothesized that patients, triple negative breast cancer patients, that do show activation of these targeted proteins would be those that might respond to neratinib therapy. Next slide. So this graph here shows you on the x-axis activation of the EGFR receptor, and on the y-axis activation of the ERB2 receptor or the HER2 receptor. And those patients in the upper right-hand quadrant, as delineated by the dashed lines, and those that are in the green squares, are patients that were treated that responded to this disease, to neratinib treatment in this trial. And this proved our hypothesis was correct. Next slide. And what we found is that if we were to then apply these biomarker positive rates, those patients that did have activated levels of these proteins within their tumors, that we had a much higher response rate to neratinib, as opposed to patients that all the triple negative patients, regardless of their activation state. So we went from a 37% response rate up to a 77% response rate if we looked at the biomarker activity within that arm. And so if we are ultimately able to independently validate these observations, these candidate biomarkers could be used to more accurately identify triple negative patients that might respond to this class of therapies. Next slide. And so finally, I have to acknowledge my colleague, Sella Gallagher, who's been my partner in conducting these I-SPY2 analyses since the beginning of the trial in 2010. So I really want to thank her. She's been an excellent colleague, and as well as acknowledge there on the right side of the slide, our colleagues at the University of California, San Francisco, where the I-SPY trial is headquartered. And those are our colleagues there on the right. So thank you very much. And our next speaker is Dr. Mary Elena Parabon. And Mary Elena came to our group almost 15 years ago on a research fellowship from Italy, where she received her medical degree and also trained as a surgeon. In 2019, she was the recipient of a Fulbright scholarship, of which she used to spend her time teaching in Italy. And that's a very prestigious honor. We are very fortunate that after arriving in our laboratory, she chose to stay with us. And she is now a newly appointed tenure track associate professor in the School of Systems Biology here at George Mason. And she's going to tell us about her work on metastatic breast cancer.
Thank you so much, Julie, and good afternoon to everyone. I just want to take the last 5-7 minutes of the time today to talk to you a little bit about the work we are doing on metastatic breast cancer. And I would like really to take this time to stress a very important collaboration that we have established with the Side Out Foundation, which is a local organization here, born and grown in our community. So really talk about those innovative collaboration opportunities that we have. Next. So I think that Lance has already given us a little bit of background about metastatic disease. So those are tumors that have not only learned to invade the local microenvironment, but also to travel to distant organs, like, for example, bone, the liver, the lung, etc. And unfortunately, the development of a metastatic lesion is usually the cause of death or associated of death for cancer patients. So finding novel ways to treat cancer patients that develop metastatic lesions still remains a very important public health priority and also a very important research question. Next. And so in the last 10 years or so, we really have seen a paradigm shift in the way that we treat cancer patients, in the sense that by using new technologies, we have learned more about the biology of tumor, and we have come to realize that tumors are highly heterogeneous. So even patients that have a very similar tumor from a clinical perspective or a pathological perspective might be driven by completely different molecular events. And so understanding this heterogeneity has really led us to appreciate and embrace the opportunity to develop precision medicine or personalized treatment for cancer patients. And so in the new models or the new clinical trials that have been developed over the last few years, and here I have just an example about the NCA match trial, we are actually really using molecular information routinely in our diagnostic and in the selection of different type of therapeutics for our cancer patients. And what we are actually doing in this new clinical trial is to use and identify molecular alterations that are responsible for the development and the progression of individual tumors. And we use this information to actually tailor treatment to individual patients. And once we identify those molecular alterations, then we use this information to really identify either FDA approved or experimental compounds that we can treat our patient with in a more effective way. And this new model, this new clinical trial model is actually quite effective because it allows us to provide personalized or tailored treatment to each individual patient. Or every patient that entered the trial is treated, so none of the patient is allocated to a control group where they would simply receive a placebo, which often is a deterrent, especially for metastatic breast cancer patients for entering those innovative clinical trials. And what we have seen over the year is that most of those trials rely heavily on genomic information. So they mostly look at changes in the DNA. But unfortunately, when we only use genomics, this type of approach allows us to deliver precision medicine to only approximately 20 to 30% of cancer patients. So we still have a big percentage of patients that cannot really benefit from this type of approach. Next slide. And so here is really where our collaboration with the Side Out Foundation have given us a unique opportunity to test a new model. and develop clinical trials where we use what we know as a multi-omic approach. So where we combine different cutting edge technologies to look at changes at the DNA level, RNA level, protein, and specifically in our laboratory, as Julie and Jenny have mentioned before, look at signaling network and alteration of signaling network that can be used as therapeutic targets. And just quickly to give you an overview, the Side Out Foundation is a nonprofit organization that was founded and is still located here in Fairfax. So it's really something that has been generated in our community to try to answer important questions and trying to improve treatment for patients that are affected by metastatic breast cancer. Next slide. Here, I just have a quick overview of the technologies that we use in our laboratory. Of course, we became part of this big foundation effort because of the unique technology that Dr. Petricone and Liotta have developed over the year. Next slide, please. And so far, we have worked with the Side Out Foundation on 3 different clinical trials. The first and the second trial were really pilot analyses aiming at exploring whether this approach that we wanted to test, so this multi-omic molecular approach, was actually a positive approach for helping physicians select the most effective treatment for our cancer patients. And the first 2 clinical trials were nationwide clinical trial. We had 4 different hospitals where patients were enrolled across the national territory, and 3 to 4 laboratories that were covering different aspects of this molecular profiling using, again, genomic, proteomics, transcriptomic, and then our signaling network profiling approach. Next, please. So the Side Out 1 and Side Out 2 trial, as I mentioned before, was really trying to answer in very basic question. And the first question is, if we use all of these technologies, if we really try to dissect at a multi-omic level those tumors, can we provide oncologists information that are useful for selecting treatment for metastatic breast cancer patients? And are those treatments effective or not for our patient? And in both clinical trials, we have demonstrated that for more than 50% of patients that have already received a large number of previous treatment, this approach is very beneficial. We have also demonstrated that by using this multi-omic approach, we can help a physician determine the best course of treatment in more than 90% of the patients. So we are really increasing the pool of patients for which precision medicine becomes available compared to genomic analysis alone. And the other very important aspect that emerged from those first 2 clinical trials is that we can do this on a national level, and we can provide physicians with information that are useful for selecting treatment in 15 business days. So this model that we are proposing is actually something that can be used in everyday clinical practice, not only in university centers, but also can be offered to patients that are treated in community hospitals. Next. So now we are working on the Side Out 3 trial. This is an ongoing trial. I will not discuss this at length because I would like to give you time for some questions, but one of the aspects I would like to stress about this trial is the number of institutions that have come on board to help us realize this third phase of the trial. So definitely a trial that has a much higher impact nationwide, and this is just a map indicating the different universities and cancer centers that have actively been involved in enrolling patients for this new effort. Next slide, please. And then I would like just to share one more aspect that has been emerged in the last 2 years through the work that we have been doing with the Side Out Foundation, which is the development of a new bioinformatic portal where all the information from the clinical trials are stored and made available to the scientific community, to cancer patients, and to their physician. And so this was one of the challenges that the Side Out Foundation brought to us. And thanks to a collaboration between the College of Science and the College of Health and Human Service, we have now developed this database that's available for everybody to access, where for each patient that was enrolled in the Side Out clinical trial, there are over 700 variables collected, including all their molecular information, their clinical data, their treatment information, and outcome. Next, please. And the very last slide, I just would like to acknowledge everybody that has made this possible. I'm just a spokesman tonight. This is not just my work, it's an effort of a very big community of scientists, volleyball players, coaches, physicians, coming together to really realize this big dream. And again, I would like to stress the importance of how all of this was possible really by engaging with a foundation that is here in our community. And so again, the importance for us as scientists to embrace challenges and questions that the community has so that we can really move forward and try to answer very important clinical and research questions. And with this, I close my presentation. I know that have gone fast. I was just trying to buy some time. So if there is any question, I'd be happy to answer them.
So thank you very much, Ginny, Julie, and Maria Elena for these very exciting and insightful presentations. We have a few questions in the Q&A box. So in the interest of time, what I'll do is I'll ask one question of each of you, and then we can move to the closing so we don't run over time. So for Ginny, so here's a question that's in the box. So we've heard a lot of comments in the news about chloroquine and COVID-19. Why does chloroquine work as a treatment for breast cancer but not COVID-19? Is it dangerous, toxic? Does it have side effects?
So chloroquine works for breast cancer because it specifically targets the autophagy pathway. And as I mentioned in my talk, chloroquine has been used for years worldwide in millions of patients for lupus, for rheumatoid arthritis, as a malaria preventive. COVID is a completely different type of disease and has a completely different mechanism of action, which is why chloroquine is not effective for COVID, but it is effective for cancer. We've also used chloroquine in other types of cancers, specifically pancreatic cancer. And the important thing to know about any drug is that, yes, there can be side effects, and depending on each individual person, particularly in a clinical trial, they're evaluated for eligibility in the trial to rule out any, for example, with chloroquine, any heart arrhythmias or any eye retinal problems that could make them ineligible to use that particular drug.
Thank you. Wonderful. Thanks, Ginny. So a question for Julie. What directions will the I-SPY trial be taking in the future?
So there's some very exciting developments in the works for the I-SPY trial consortium. Starting next year, the trial is going to move on to what we call I-SPY 2.2. And the structure of the trial is going to change a little bit and address sort of one of the big issues in cancer therapy in general, breast cancer specifically, surrounding the toxicity of standard chemotherapy drugs and the need to try to avoid those toxicities. And so the new structure for the trial will be that experimental agents will be given as monotherapy or alone by themselves to test their efficacy. And patients will be allowed, if they show a very limited response within weeks of starting these therapeutic agents, if their tumor is not responding, they'll be allowed to move on to other drugs that have actually been successful in the I-SPY 2 trial as a second option. And so those are some of the directions that we're going to be going in, and those should be starting next year.
Great. Thanks very much. And then one question for Maria Elena. We often see these clinical trials being designed and carried out by university and cancer centers. Do you think this approach can also be offered in community hospitals at some point?
Yeah, so that was one of the questions that we tried to answer with the Side Out clinical trial. So we had those multi-institutions that were enrolling patients, and especially in the last, the Side Out 3 trial, we had university hospitals, we had cancer centers, but also some of the community hospital, one here in Fairfax, and the community hospital that work around university hospital, being able to enroll patients and so really show that this is a workflow that can potentially be offered to a broad number of different institutions, including the community hospital. And that is extremely important, especially in the metastatic setting, because the vast majority of metastatic breast cancer patients are followed and treated in community centers. So it is really, really important to develop those workflows that will transcend and that can really be accessible to all our patients.
Yeah, wonderful, wonderful. There's one more question that came in, and I don't know if it's who is it addressed, so I'll just ask it and ask any of you to take it. Is there a change in the pathophysiology at the cellular molecular level between in situ breast carcinoma cells and metastatic breast carcinoma?
I can take on a little bit the question. I think Jenny probably can give a little bit more of a background from the in situ to the invasive cancer, but for sure what we see, and we have worked on this in the past few years, is that when you look at even at the primary tumor and the metastatic lesion, at the molecular level, those two lesions can be extremely different. So there is definitely a selection of specific molecular event that allow the tumor cells first to invade locally, to survive in the systemic circulation, and then to colonize and grow in a different organ. And also the organ in which they grow can modulate the signaling network that the tumor strongly relies on. So there is definitely a kind of like an evolution and a selection for specific subclones.
Okay. Well, great. Well, since we're running a little behind, and I want to keep this running as closely to time as possible, and this is not unusual, particularly when we have such exciting topics. I think I'll leave it there. I want to just offer some closing comments. First of all, thanks to our host in our panel for this fascinating and enlightening discussion. I think it's, I want to just to note that even while this session opened with 3 males, including myself, these 3 groundbreaking trials are led by women. And I'm very proud to say that the College of Science is truly exceptional in that over 61% of our undergraduate student body is female. And roughly 50% of our graduate students are female. And these are exceptional statistics for a College of Science anywhere in the world, and certainly in the US. So I'm really proud of that. And as a college, we're deeply committed to diversity, diversity of gender, of race, of ethnicity, of culture, diversity in the broadest defined sense. Every study that we've seen in terms of the impacts of participation of the diverse group of people in science points to the fact that you get better results, you get results that are more impactful, and you involve a wider diversity of people. So this is really something that's important to us, and I'm really proud of our college and of our researchers in this respect. And today was just one example of many we could offer. In closing, I would like to echo our thanks as given earlier to the Beck Foundation for their ongoing support for the College of Science and for George Mason University as a whole. The Beck Foundation faculty fellowships provided seed funding for some of the amazing research we just learned about. And thank you, friends, alumni, colleagues, for joining us this afternoon. We hope to see you again on October 29th, when we'll learn more about one of the fastest growing programs in the College of Science, with scientists from our forensic science program who will discuss our plans for a body farm just time before Halloween. So thanks again. Wonderful to be here with you. And thanks to everyone for attending, for organizing. It's just great. Thank you.